Please welcome Terence Lee, Hong Kong Polytechnic University.
I'm Terence Lee, professor in the Department of Applied Biology and Chemical Technology in Hong Kong Polytechnic University. Here, I will recommend Ynno, a university spin-off biotechnology company dedicated to address the unmet clinical needs of new antimicrobial agents. And the company used their proprietary and unique technology by targeting the protein-protein interactions as a novel drug targets, which revolutionized the current treatment strategies for antibiotic-resistant bacterial infections. Their drug development platform is also accelerated by the AI program developed in-house. To date, multiple pipelines has been developed, and I believe more to come. Our university's motto is to learn and to apply for the benefit of mankind. I sincerely believe that Ynno can significantly contribute to the antimicrobial treatment, human health, and also the society. Thank you.
Breaking the wall to novel antibiotic candidates. Xiao, Ynno Med.
Angela was 28 when she was admitted to our hospital earlier this year. She needed cardiac surgery. However, this operation could not take place because doctors discovered Angela was carrying a dangerous chest infection by multi-drug-resistant Pseudomonas aeruginosa. Although her doctors tried everything they could, Angela did not survive. What makes this story even more heartbreaking is that it happened in a fully modernized hospital in Hong Kong, one of the world's largest metropolitan cities. Our doctors have access to all the antibiotics available, but none of them were effective against the bacteria that claimed Angela's life. It was a battle we could not win. Angela's case is not an isolated incident. In 2019, if this thing responds to me, there were 5 million such sad cases. It's a global crisis. A recent UK governmental review estimated that if we don't take action now, by 2050, if this thing responds again, we will have 10 million deaths attributable to antibiotic-resistant infections with over $100 trillion medical expenses incurred. We are witnessing an alarmingly increase of antibiotic resistance while there were no antibiotics. Since 1980, no new drug, no new class of antibiotics were developed. This is a perfect storm, and we have to urgently address it with new antibiotics, a lot of them, preferably. The traditional way of drug development is looking for enzymes, less tiny little machines that support bacteria's life. Traditional antibiotics, they target the active site of the enzyme and jamming these machines. However, bacteria, they're incredibly adaptive. So if they mutate the enzyme can still function while they become resistant to the medicine. It was a game of cat and mouse that bacteria are winning. In Ynno, we developed first-in-class technologies to target protein-protein interactions in the essential bacterial machineries. Our technology mimics one of the proteins and preventing it binds to its protein partners. In this way, we're disrupting the machines and causing bacteria to die. In another word, instead of turning off the machines, Ynno we're trying to break it apart. So even bacteria try to outsmart us by mutating one of the proteins we target. Of course, it can initially, I don't know why this jumps around, but of course, it can try to escape our antibiotics initially. However, since the mutation happened at the interface of the protein-protein interaction, it makes the protein harder to interact with partners, and in this way, bacteria will still die. Based on this technology, we targeted a specific bacterial machinery, that's transcription machinery. It's responsible to making RNA based on its template DNA. We have discovered and developed 8 projects, 8 new class of antibiotic candidates. And 3 of our leading projects, they already proven safety and efficacy profile using animal models. This one particular molecule shown here is effective against Pseudomonas aeruginosa, a bug that is stubbornly resistant to many of the existing antibiotics. This is the very clinical isolate that caused Angela to die. This particular compound is sitting in the fridge of the same hospital where Angela passed away. The potential to save lives is within our hands, but we need your help. It's not a drug yet. It really pains me and fuels my determination to stand in front of this audience and say, we need money. We need financial support to further our pipelines. And UK, Hong Kong government, they promised that, let's test infection. See, Hong Kong government promised that with every dollar investors deposit in Inno's bank account, they are going to match it with $2 of their own. With this level of support, we can pump out new antibiotics into the clinic. We work to ensure a future where no life was lost due to bacterial infections. Thank you.
On point. Great. Questions for those of you who were sleeping, you were woken up. That's good, because we're not finished yet, so let's go. Questions, please? Yes, Frank? Thank you to bring the microphone. I see you at the back. You know the process. Yes.
Honestly, RNA polymerase as a target is not really a very new target in bacterial or antibiotic discovery. What makes these compounds different to other ones? Also, the protein-protein action between the different subunits of the RNA polymerase is targeted by others.
Yeah, you're right. RNA polymerase is actually a proven target. So clinically, we have rifampicin and fidaxomicin. They're targeting the active side of this enzyme. So in Innomed, we're trying to target the essential transcription factors that regulates this process. So something like initiation factor, sigma, and other elongation factors. I also target the interface between some essential transcription factors. So, yeah, if that answers your question.
Yes, okay. Then I don't see anyone in the jury yet. So let's take the question here at the back, please, of the gentleman with the reddish. Yeah.
Do you expect that these will be held in reserve in terms of reimbursement, that they'll be held in reserve for isolates that are resistant to everything else, or do you intend to put these out as frontline antibiotics, because that obviously affects the funding stream?
Yeah, we're in the process of indication development, but well, antibiotic resistance is really a multifactorial problem, and you know, there are scientists we're up for discovery and development, and we need to work with industry, we need to work with policymakers, and we need to influence the first-line doctors for the prescription habits. And also, I think awareness is a big thing too. So I don't know if that answers your question, but this is really a multi-dimensional question. I can talk to you later with your specific aspect.
Okay, good. Next question. Anyone here? Angelica, please. Thank you.
Thank you very much for your nice presentation. Just a question on the quality of your data. Are these based on animal models, or are these competitive assays, or what does you show us?
Yeah, we have done in vitro activity assays. We have done animal models, animal infectious models. We've tested the PK. We've developed about chemical assays, about physical assays, structural assays. For the mechanistic studies, we have done the PK, tissue distribution, and pretty much we have done whatever we can with our resources so far. Thank you.
Okay. It will be last question, probably. Chaz, please. Thank you. Thank you.
Very, very simple question. Is your bottleneck only money?
I hope so. That would be nice. But you know, drug discovery is really, really a long and a risky process. And we're trying pretty much everything we can. And I didn't really touch much today as we develop our own in-house AI algorithms, and we use computer-based drug discovery and design for accelerating the whole process. So we do consider a lot of the downstream problems. I talked to industry at the first place. A lot of the industries, we care a lot about the mechanistics, the activities, but industry, they care more about the cost. Am I going to make money? I talk to clinical physicians as well. They care about the safety of the drug. So as I said, it's really a multidimensional problem. Yeah, we need more money to push it forward.
Okay.
Thank you.
You're welcome. Thank you very much. We'll finish